Good morning everyone. Today we discuss different types of protein present in actin myofilaments, myosin myofilaments and muscle contraction. proteins present in the actin and myosin myofilaments. Actin filament contain three types of proteins: actin protein, trophomyosin and trophonin. Actin protein contain two filamentous actin. It is one filamentous actin, it is another filamentous actin, and each filamentous actin is a polymer, contains several monomers. Each monomer is a globular protein, circular protein. So actin protein contain two filamentous actin. Each filamentous actin is a polymer. It contains several monomers. These monomers are globular protein. Globular protein means a circular protein. Next one, trophomyosin. Like actin protein, trophomyosin also contain two filamentous strands. The two filamentous strands of trophomyosin attached to throughout the length of actin protein. Next one, trophonin. Trophonin is the complex protein, contain three subunits: trophonin T, trophonin C and trophonin I. With the help of TNT subunit, trophonin bind to trophomyosin. The subunit of trophonin to which calcium ions bind is known as trophonin C. So with the help of TNT, trophonin bind to trophomyosin. As a result, trophonin-trophomyosin complex is formed. The subunit of trophonin to which calcium ions adhere is known as TNC, trophonin inhibitor. The trophonin inhibitor try to cover the active sites. So these are the active sites, exposed part of the actin protein, active sites. Meromycin head. Meromycin is the monomer of mycin protein. The meromycin head, with the help of actin binding site, attaches to active sites of actin protein. In the absence of calcium ions, observe, it is example. In the absence of calcium ion, trophonin-trophomyosin complex cover or mask the active sites. Now the meromycin head unable to attach to active sites. So with the help of meromycin head, the mycin filament attaches to actin filament during muscle contraction. So in the absence of calcium ions, trophonin-trophomyosin complex cover the active sites or mask the active sites. Now the mycin filament unable to attach to actin filament. That's why trophomyosin, trophonin are known as regulatory protein. In the presence of calcium ions, trophonin-trophomyosin complex move away from the active sites. Now active sites are exposed. Whenever the active sites are exposed, mycin filament attaches to actin filament. In the absence of calcium ions, trophonin-trophomyosin complex cover the active sites. Now the mycin filament unable to attach to actin filament. That's why trophonin-trophomyosin are known as a regulatory protein. So it is a hypothetical diagram. Generally, all the three proteins are present in the actin filament in this manner. So trophonin attaches to trophomyosin here and there at regular intervals, not continuously throughout the length of the trophomyosin. Trophomyosin attaches to actin protein throughout the length. Three proteins present in the actin filament: actin protein, trophomyosin protein, trophonin protein. Actin protein contains two filamentous actin. It is one filamentous actin. It is another filamentous actin. Each filamentous actin is a polymer, contains several monomers. This monomer is globular protein, spherical protein. Next one is trophomyosin. Trophomyosin contain two filamentous strands. The two filamentous strands of trophomyosin attaches to actin protein throughout the length. Another protein is trophonin. Trophonin is the complex protein. It contains three subunits: TNT, TNC, TNI. With the help of TNT, trophonin. With the help of TNT, trophonin attaches to trophomycin. As a result, trophonin-trophomycin complex is formed. In presence of calcium ions, so what is TNC? The subunit of trophonin attaches to calcium ion is known as TNC. TNI means inhibitor. The subunit of trophonin prevent the binding of mycin filament to actin filament. So that is trophonin I. In the presence of calcium ions, trophonin-trophomycin complex move away. For example, in this diagram, so not visible. So maybe move away. Trophonin-trophomycin complex move away from the active sites. Active sites are exposed. In the absence of calcium ions, trophonin-trophomycin complex cover the active sites. So that means in presence of calcium ions, trophonin-trophomycin complex uncover the active sites. In the absence of calcium ions, they mask the active sites. That's why trophomycin and trophonin are known as regulatory proteins. So it is a hypothetical diagram. For our convenience only, I drew separately. Actually, all the three proteins are arranged like this in the absence of calcium ions. In the presence of calcium ion, trophonin-trophomycin complex move away from the active sites. Next one, mycin filament contain only one protein, mycin protein. Mycin protein contains 6 polypeptide chains, 2 heavy polypeptide chains, 4 light polypeptide chains, 1, 2, 3, 4. 2 heavy polypeptide chains and 4 light polypeptide chains. On one end, the 2 heavy peptide chains coiled, as a result tail is formed. In another end, each heavy polypeptide chain folded, as a result head is formed. So mycin protein contain 1 tail, 2 necks and 2 heads. Each head attaches to 2 light polypeptide chain. What is heavy polypeptide chain? Polypeptide chain contains several amino acids known as heavy polypeptide chain. Polypeptide chain contain few amino acids known as light polypeptide chain. So next one is mycin protein. Mycin protein contain 6 polypeptide chain, 2 heavy polypeptide chains, 4 light polypeptide chain. On one end, 2 heavy polypeptide chains coiled, as a result tail is formed. Another end, the heavy polypeptide chain folded, as a result neck is formed. So mycin protein contain 1 tail, 2 necks and 2 heads. Each head attaches to 2 light polypeptide chains. So monomer of mycin protein is meromycin. 200 to 300 meromycins are present throughout the length of the mycin protein. 200 to 300 meromycins are present throughout the length of the mycin protein. So mycin filament contain only mycin protein. Mycin protein contains 6 polypeptide chain, 2 heavy polypeptide chain coiled with each other. On one end, tail is formed. Another end, each heavy polypeptide chain folded, head is formed. So each mycin protein contains 1 tail, 2 necks, 2 heads. Head connected to 2 light polypeptide chain. Throughout the length of mycin protein contain 200 to 300 monomers. They are known as meromycin. Each meromycin contain 1 head, short arm and tail. Head and short arm contain several amino acids. That's why it is known as heavy meromycin. And tail contain few amino acids. That's why tail is known as light meromycin. Head contain 2 binding sites. With 1 binding site bind to ATP molecule, that is known as ATP binding site. With another binding site, head of meromycin attaches to active sites of actin protein. Active sites of actin protein. So types of proteins present in myofilaments. So actin filament contain three types of protein: actin, trophomycin, trophonin. Mycin filament contain only one protein, mycin protein, and monomers of mycin protein are meromycins. So 200 to 300 meromycins are present throughout the length of the mycin protein. So Z layer. Now we discuss how the meromycins are arranged. It is the mycin filament. Mycin filament contain mycin protein. Tail remain in the mycin filament. Short arm and head exposed. Exposed parts of the mycin protein. So meromycin known as cross arm. Tail remain in the mycin filament. Short arm and head exposed. Exposed parts are known as cross arm. Observe, it is the mycin filament contain mycin protein. We already discussed each mycin filament or mycin protein contain. Each mycin protein contain 200. So each filament contain 200 to 300 meromycin. We assume 300 meromycins are present. 150 meromycins with the help of head attaches to actin filaments of one half of the I band. Another 150 attaches to meromycin heads attaches to actin filaments of another half of the I band. You already it is the sarcomere, Z layer, half of the I band, A band and half of the I band. Mycin contain 200 to 300 meromycin. We assume 300 meromycins are present. 150 meromycins with the help of head attaches to actin filaments present on one half of the I band. The remaining 150 meromycin attaches to actin filaments of half of the I band. Later, the meromycins pull the actin filament towards the M line. So, due to pulling, what happened? Actin filament slide over. Now, only in the peripheral part only actin filament slide over mycin. Due to continuous pulling, the actin filaments slide over mycin filament. So this theory is known as the sliding filament theory. So each mycin contain 200 to 300 monomers. The tail remain in the mycin, short arm and head exposed, and exposed parts are known as cross arm. Cross arm attaches to active sites. As a result, cross bridge is formed. Cross bridge is a connection between actin and mycin filament. Actin filament contain mainly actin protein, mycin filament mainly contain mycin protein. These two proteins unite through cross bridge. As a result, actomycin complex is formed. So 50% meromycins attaches to actin filaments of one half of the I band. The remaining 50% meromycins attaches to actin filaments of another half of the I band. The meromycins pull the actin filament towards the mycin. As a result, actin filaments slide over mycin filament. That's why it is known as sliding filament theory. Now we discuss sliding filament theory. Triad system play a key role during muscle contraction. It is a myofibril. You all know in a muscle cell, several myofibril. It is a muscle cell. In a muscle cell, several myofibrils are present parallel to the longitudinal axis. The plasma membrane of the muscle cell, sarcolemma invaginate. Invagination means fold inwards. As a result, T-shaped tubule is formed. T-shaped tubule is formed near the Z layer. The cisternae present immediately above the T tubule, the cisterna present just below the T tubule. These three structures together known as triad system. triad system play a key role in muscle contraction. In mammals, the triad system is present near the I band and A band. Near the I band and A band. In mammals, the triad system is present near the between the I bands and A bands. So, in a sarcomere, it is the sarcomere. It is the I band and A band, one triad system. It is the A band and I band, another triad system. So, in mammals, triad systems are present between I band and A band. How triad system is formed? Sarcolemma invaginate, fold inwards. As a result, T-shaped tubule is formed. The T-shaped tubule is formed between the I band and A band. The cisternae, you all know endoplasmic reticulum of the muscle cell known as sarcoplasmic reticulum. The sarcoplasmic reticulum release calcium ions. So, the cisterna present above the T tubule and cisterna present below the T tubule. These three structures together known as triad system. Triad system play a key role in muscle contraction. How triad system is formed? The sarcolemma invaginate between I band and A band. T-shaped tubule is formed. Cisterna present above the T-shaped tubule and cisterna present below the T-shaped tubule. These three structures together known as triad system. Sliding filament theory successfully explained the process of muscle contraction. Hugh Huxley and Gene Hansen proposed the sliding filament theory. Hugh Huxley and Gene Hansen proposed the sliding filament theory. According to this theory, actin filament slide over myosin filament during muscle contraction. First one is muscle excitation. The central nervous system via motor nerve release the message between motor neuron and the muscle. The empty place present between motor neuron and muscle is known as neuromuscular junction or motor end plate. From the motor end plate, the message transferred to T-tubule. From the T-tubule excited and transfer the message to the cisternae. Cisternae excited, release calcium ions into the sarcoplasm. Now TNC subunit of troponin bind to calcium ions. Now the troponin tropomyosin complex move away from the active sites. As a result, active sites are exposed. It is the myosin filament and meromyosin. So now whenever active sites are exposed, meromyosin head in the absence of ATP molecule, means in the presence of ADP molecule, attaches to active site. As a result, cross bridge is formed. Myosin filament and actin filament connected by a cross bridge. What is cross bridge? Cross arm plus active site of actin protein. You already know. Tail remain in the myosin filament. Short arm and head together known as cross arm. Cross arm connected to the active sites of actin protein. As a result, cross bridge is formed. Once again, sliding filament theory successfully explained the muscle contraction. Hugh Huxley and Gene Hansen explained the sliding filament theory. Central nervous system send message to muscle via motor neuron. Motor neuron release the message in the neuromuscular junction. From the neuromuscular junction, the T-tubule receive the message. T-tubule excited, transfer the message to cisternae. Cisternae excited and release calcium ions into sarcoplasm. Now calcium ion bind to TNC subunit of troponin. As a result, troponin tropomyosin complex move away from the active sites. Now the meromyosin head in the absence of ATP, means in the absence of ATP means in the presence of ADP, bind to myosin, bind to actin protein. So later actin protein. So now the meromyosin head attaches to actin protein. Now meromyosin pull the actin filament towards the, so like this here. The meromyosin head pull the actin, for example it is the meromyosin head, pull the actin towards the myosin filament. It is known as power stroke. Power stroke. The meromycin head pull the actin towards the myosin filament is known as power stroke. After power stroke, from the ATP site, ADP is separated. Later, head again regain its original position known as recovery stroke. After recovery stroke, ATP molecule attaches to ATP site. At the time, meromycin head detached from the active sites of ATP. Now the head of meromycin head act as ATPase enzyme and converts ATP into ADP plus PI by utilizing this energy. So ATP already converted into ADP. By utilizing this energy, it attaches to another active site of the same actin filament, and the process is repeated. This process is repeated. Due to repeated contraction, actin filaments slide over myosin filaments. As a result, I band and H zone decrease or disappear. No change occur in the length of A band. It is known as muscle contraction. Once again, sliding filament theory, or with the help of diagram. One more diagram. Look on the board. Sliding filament theory proposed by Hugo de Vries and Hugh Huxley and Jean Hansen proposed the sliding filament theory. Sliding filament theory means actin filament slide over myosin. Sliding filament theory. Central nervous system send the message to muscle, then only muscle undergo contraction. The motor neuron. The central nervous system transfer the message via motor neuron. Motor neuron release the message in the neuromuscular junction. From the neuromuscular junction, T-tubule receive the message, excited, transfer the message to cisternae. Cisternae excited, release calcium ions. It is known as muscle excitation. Due to muscle excitation, abundant number of calcium ions present in the sarcoplasm. Now TNC subunit bind to calcium ion. Now trophonin-tropomyosin complex move away from the active sites. In the absence of ATP, Meromycin head attaches to active site. Later, pull the actin towards the mycin. It is known as power stroke. After power stroke, ADP molecule attaches to ATP binding site separated. Now it regains its original position known as recovery stroke. Later, new ATP molecule attaches to ATP site. Whenever ATP molecule attaches to ATP site, the meromycin head detached from the active site, and the whole process is repeated. What happened again? Now the meromycin head act as a ATPase enzyme. It hydrolyzes the ATP into ADP. By utilizing this energy, meromycin head attached to the active site. Later pull the actin towards the mycin, power stroke. This process is repeated. This process is repeated. Due to repeated contractions, what happened like this? It is the meromycin. The meromycin pull the actin towards the mycin. The meromycin pull the actin toward the mycin. As a result, observe it is the relaxed sarcomere. It is contracted sarcomere. So it is the length of I band, but here length means I band decreases, and it is the H zone. Here H zone decreases. In a maximum contracted, observe, sarcomere contain Z layer, A band, Z layer, means I band decreases, H zone disappear. Here H zone. In a relaxed sarcomere, I band, H zone. In contracting sarcomere, I band, H zone decrease. In a maximum contracted sarcomere, I band and H zone disappear, but no change in the length. For example, one half of the I band 2 millimeter, another half of the I band 2 millimeter, A band 10 millimeters, total sarcomere length 14 millimeters. In a maximum contracted sarcomere 10 millimeters only. So that is A band. So half of the I bands disappear. So during muscle contraction, 14 millimeters sarcomere converted into 10 millimeter sarcomere. In this manner, the muscle sarcomere shorten, automatically muscle shorten. So muscle contraction. So tomorrow, once again, I revise the sliding filament theory. So tomorrow I draw small diagrams and simultaneously we discuss sliding filament theory and muscle contraction. I repeat once again, sliding filament theory.